So good evening, all of you. Ma.
Good evening, sir.
Good evening. So yesterday we stopped somewhere in the topic of the basic genetic concepts. So we will continue in this class. So yesterday we discussing about this one, ma. We stopped here about the extra chromosomal inheritance or cytoplasmic inheritance or extranuclear or uniparental inheritance. So all are represent these are inheritance is controlled by the DNA present in the chloroplast and mitochondria. So the Corens is considered as the father of the cytogenetics, because here the chloroplast and mitochondria comes under the cytology. We will read in cytology, under the cell organelles. But these two organelles having the DNA, and these are involved in the inheritance of the organisms, in some organisms, not all organisms. So that's why these two are involved in the cytoplasmic inheritance or extra chromosomal inheritance or extranuclear inheritance or uniparental inheritance. So this branch of cytogenetics was started by the work of the Corens. So that's why he was considered as the father of the cytogenetics, or founder of the cytogenetics. So you keep remember this one, the chromosome number. He has done. He has started this work on the. He has started work on this the Miraculous Jalapama. It is also called as 4 o'clock. So the chromosome number is 2n is equal to x is equal 58, So you keep remember this chromosome number of this Miraculous Jalapama. Sometimes they will ask like chromosome number of the organisms also in genetics. And coming to the Hugo de Vries, as all of you know, he was the father of the mutation breeding. He has started before the mutation. He also worked on the inheritance pattern. So he has worked on the inheritance pattern, and he got the same result as Mendel got, and he has approved that the Mendel was the correct, and Mendel laws are the universal. So after that, the another contribution of the Hugo de Vries is he proposed the mutation theory, and he was considered as the father of the mutation breeding, and he has worked on the Oenothera lamarckiana. Oenothera lamarckiana. It is also called evening primrose. So the chromosome number is 2n is equal. So the chromosome. So the chromosome number is 2n is equal to x is equal 14. So for Miraculous Jalapa, 2n is equal to x is equal 58. For Oenothera lamarckiana, 2n is equal to x is equal 14. And this chromosome number is you can see in the garden pea also having the 14 chromosome number, and wheat also having the 14 chromosome number, and oat also having the 14 chromosome number. 2n is equal to x is equal 14. So you can see in the wheat, in oat, and garden pea also. And coming to the Johnson W. So he was the famous in plant breeding, but he has used the some terminology related to the genetics and plant breeding, that is the genotype and phenotype. He has written one book also. These are elements of an exact theory of the heredity. So in this book, he has mentioned these two terms, genotype and phenotype. This after the publish of this book, these two terms are popular in the field of the genetics. So he has proposed he has proposed the pure line theory also. He has given the term pure line also, and he has given the term of gene, genotype, and gene also. and he was proposed the purulent theory, working on the common bean, princess variety of common bean, that is a Phaseolus vulgaris, this one ma. And he also worked on the barley also ma. Actually, he did work on the barley and common bee, and he proposed the purulent theory. But we will study in plant breeding, we will study only the purulent theory on the princess variety of the common bean only. But he has done on the work on the barley and common bean. So this is the bean Phaseolus vulgaris, and chromosome number is 2n is equal to x is equal to 22. And which pulse crops having this chromosome number? 2n is equal to x or 22. PGNP also having red gum, yes ma. So red gum also having the 22 chromosomes. And T.H. Morgan. So he was the most important person in the field of the genetics and field of the conventional genetics. So he was considered as the father of the experimental genetics because he has done extensive work on the Drosophila, and he has proved the Mendel laws as well as chromosomal theory of inheritance. He proved this both theories because Mendel has proposed theories based on his work, but there is no experimental proof for the Mendel laws. Only he has proposed the laws based on the mathematics, because up to Mendel, there were no scientists who are used the mathematics in the field of the biology. In the field of biology, because genetics also comes under the biology, and this botany, zoology, all are comes under the biology. But only Mendel has started the use of the mathematics to explain the biological theories. But there is no proof. Mendel has only imagined there are some factors are responsible to governing that character. He don't know what is that factor and how that factor made up. Means it is made by the DNA. He doesn't know it is present in the nucleus, and nucleus present in the cell. He doesn't know anything about that background of the character. The background of the characters are controlled by DNA. That DNA present in the nucleus. So he doesn't know. The background of the character. He knows only the some factors that are present in the individual that are responsible for the character. He doesn't prove that how the character will where it is present, and how they will transfer, and how it is transferred. He doesn't know about that one. So, but after the experiment of the Morgan, he has done the experiment on the transmission of white eye in Drosophila. So after this experiment, he has given the conclusion about the Mendel laws are accepted, and Mendel laws are followed like this, and we have to accept a chromosomal theory of inheritance also proposed by Sutton and Boveri. So that's why he was the most important in the genetics. T.H. Morgan. And he has extensive work on the Drosophila. The chromosome number is 2n is equal to x is equal to 8. So Drosophila having the chromosome number of 8. And he has done on the work on the Drosophila, and he observed the transmission of character of the white eye color. That is in Drosophila generally having the red eye color. It is a dominant one, and he suddenly observed another another alternative color, that is the white colored eyes of the Drosophila. So here I want to mention one thing, ma. Generally, we will use in genetics. We will use the we will represent the alleles in two ways, ma. In genetics, we will represent the allele in two ways. So in genetics, we will represent the alleles. Alleles means the alternative form of the gene. So one gene you can represent it by the two alleles. It is represented by the two alleles. That is the one is dominant allele, another one recessive allele. That gene may be expressed in homozygous dominant or homozygous recessive or heterozygote form. So what are the two systems are to represent the allele of the gene in the genetics? The first one is the capital and small system, small letter system. Capital and small letter system. Capital and small letter system. How we will represent this one means, for example, if flower color is there, if you will see the flower color in P, there are the two flower color in P. One is the red, another one is the white. So the red color is the dominant, so we will represent in the capital letter, and the white color is, so red color is dominant, so that's why we will represent the capital R, capital R, or capital R, small r, capital R, small r. So generally we will represent, it is controlled by the dominant allele. We will represent with the dominant allele. Coming to the recessive form of the character, that is the white, we will represent by the recessive one, that is the smaller. So this is the one system we will represent the allele of the gene in genetics, ma. The another system also is there, ma. That one is letter and symbol system. Letter symbol system. So you should remember this this both, ma, because sometimes you can see this one, ma. Sometimes you can see this one, capital and small letter system, and sometimes you can see this letter symbol system. Letter symbol system means here we will see the we will represent the alternative form of the gene by the letter and symbol. Means one dominant one. The dominant will represented by, if you will take the any character, for example, eye color in Drosophila. Generally the Drosophila having the two alternative color, one is red, another one is white. Generally in the system of the capital and small letter system, we will represent by capital or smaller, but here you have to choose, ma. Morgan represented by capital W and capital W plus, small w and small w plus. But you have to your choice, ma. So you take generally we will take the capital letter, ma. So we will represent by capital R plus, that is a superscript, ma. This plus should be superscript, and the recessive form of the allele will represent by the only that capital letter. In some time we will represent with the small, in some cases small, and the dominant form of the character will represent by small with plus. So where the plus is present, that is the dominant, that is the wild type. So you should remember this one, ma. Where the plus is present, that is the dominant form. It may be superscript to the dominant allele or recessive allele. If plus is there, that is the dominant form. So these two systems generally we will use in the genetics for the representation of the alleles. So these are the basic thing that's why I am mentioning here. So you should remember this one. It will not ask in the examination, but for your understanding and your, I'm mentioning here only. So Mendel has worked on the eye color of the Drosophila. He has observed generally in Drosophila the red color is generally you can see, but he was suddenly observed the white eye color in Drosophila. He was suddenly observed. So he has thought how it will come, how it was coming. So he has done some crosses, so Morgan explained. He has done some crosses between the white eye and red eye. So he got the red and red and red. They all he got all the reds. Among this progeny, he has done intercrossing between among F1. So he has done the intercrossing among the F1. In F2, he observed again white eye. So he observed he crossed he taken the white eye as the parent. He didn't got in the F1. Similarly, Mendel lost. So Mendel also he crossed with tall and white. He observed all tall in F1, but in F2 he suddenly got he observed he got the dwarf plant. So similarly, the Morgan also he crossed with the white eye with the red eye. He got all the red eye in F1, and after the intermating between. Similarly, Mendel has done selfing in F1 generation. Similarly, Morgan has done the intermating between among the progeny. That is intermating between the male and female Drosophila. In F2, he observed the white eye along with the 3 red eye. That is the ratio will be same. Similarly, Mendel also got the same result in same way. So by his experiment, he has approved that the Mendel was done well, and his loss we have to accept. And here you can see similar one. So by this experiment, because the eye color is present on the X chromosome, in Drosophila, the eye color is present on the X chromosome, and on X chromosome. So by his experiment, he has also concluded that the genes are present on the X chromosome, because he observed under the microscope. He observed on the microscope. Only the chromosomes are present, and these eye color are the gene responsible for this eye color is present on the X chromosome, that are transmitted to the male to female. Because in male if you want the eye color that is a white color, that should be only sufficient of the one allele only. That is the only one allele will be sufficient. But in case of the female, it should be have the 2 allele to express the white eye. But in case of the female, one allele is sufficient, because in Y chromosome there is no presence of the eye color, gene responsible for the eye color. That's why he had concluded that this the gene responsible for eye color is present on the X chromosome. So this is transmitted to the parents to progeny. That's why he concluded that chromosomal theory of inheritance is approved and it is also accepted, because according to chromosomal theory of inheritance proposed by Sutton and Boveri, worked on the grasshopper. So the genes are present according to that that theory stated that the genes are present on the chromosomes. This chromosome will carry the genes from the parents to progeny during the gamete formation by the meiosis, and it will by the meiosis during the meiosis, and by the fertilization it will pass into the progeny from the parents. So according to this theory, so by the Morgan experiment, he has concluded that this theory also approved and accepted. And coming to the CB bridge, CB bridge. So he is also one of the most important scientists in genetics, So generally, he has done on the his contribution, or he has proposed he has done on the Drosophila. He has extensive work. He has done extensive work on the Drosophila. So he proposed the genic balance theory. Genic balance theory. We will discuss in detail about this topic on the sex determination. What is mean by the genic balance theory? That means the maleness and femaleness will depends on the ratio of the autosomes by allosomes. That is the genic balance theory. So the one of this is one type of the sex determination. The genic balance theory is one of the type of the sex determination. Means the maleness and femaleness in some of the individuals are depend on the ratio of the autosomes and allosomes. And he also discovered the duplication in Drosophila in the region of the same I shape of the Drosophila. Here I shape. So Morgan worked on the eye color, but CB Bridge worked on the eye shape. So he observed the duplication. Due to duplication, the type of the eye shape is present in the Drosophila, because the 16A, that is one of the region of the gene. The 16A, one of the region of the gene that is responsible for the eye shape in Drosophila. So if the one copy of Drosophila, the region, the 16A region, one copy will give the normal eye, and two copies of 16A will give the bar eye, and 3 copies of 16A region will give the ultra bar eye. So that is the duplication. So here you can see what is the mean by the duplication. Duplication means the same copy present in the twice or thrice, or in many copies. So same one region of the you can if you'll see the copies of the gene in the form of in more than 2 copies, that is called duplication. So we will discuss in detail about this one in the chromosomal aberrations. So these are the contribution of the CB Bridgman. One is he has proposed the genic balance theory, and he identified the duplication in the region of the 16A in Drosophila. He has proposed the genic balance theory in Drosophila, and he has identified the polytene chromosome. Polytene chromosome or gained chromosome. Polytene or gained chromosome in salivary glands of the Drosophila. So this is the special type of the chromosome, because these are the this having the function, this having the structure. These two are the function and structure of these chromosomes will differ from the normal chromosomes. So about this one we will discuss in the special type of the chromosomes. That separate chapter is there. There are the Lamprey's chromosome, polytene chromosomes, and there are the A type of the chromosomes, because there are so many types of the DNA is there, A, B, C, D, Z, but we have the only B form. that is the regular one, and that will be more functioning one. So the coming to the CBBG contribution, one is the genic balance theory, another one duplication, the another one is polytene chromosome and gained chromosomes. These three are discovered or proposed or identified based on the worked on the Drosophila. And coming to the one gene, one enzyme, and the first lecture about this one I mentioned in first lecture also. So it was proposed by Beadle and Tatum, as all of you know. And here the organism is important because these two are proposed this theory, one gene, one enzyme hypothesis based on the Neurospora. This is the fungi, this is the fungi. This having the chromosome Neurospora crassa. This chromosome number is 2n is equal 14. So the Neurospora also having the 2n is equal 14, and that Garden Bee also having the 2n is equal 14, and the Enothera lamarckiana also having the 2n is equal 14. So these are the major organisms used for the genetic experiments. Neurospora, E. coli, Garden Bee, Drosophila, and in some cases grasshopper. So E. coli is most, and the Arabidopsis also used the advanced. It is used in advanced genetic research, but previously these organisms are used. So you should remember chromosome number of Neurospora also. And coming to the chromosomal theory of inheritance. So generally, I already told this chromosomal theory of inheritance was proposed by Sutton and Boveri. So these two scientists were proposed independently and simultaneously. So this Sutton was worked on the grasshopper, and Boveri worked on the sea urchins. So generally in textbook they were given in textbook. It is given that the chromosomal theory of inheritance based on the work on the grasshopper by these two scientists. But generally, the fact is the Sutton was proposed based on the work on the grasshopper. The Boveri proposed independently based on the work on the sea urchin, because this Sutton generally has done on the work on the grasshopper. He observed the chromosomes. So he observed the chromosome, the separation of the chromosomes during the gamete formation. So first he observed, first Sutton observed the separation of the chromosome during the gamete formation under the microscope, because at the time of this scientist, they having the availability of the microscope. That's why they did their experiment with microscope. So he observed the segregation, that is the separation of the chromosomes during the gamete formation, and so he has concluded that the genes are present on the chromosome. These are the carriers of the genes. So the inheritance will carried out by the chromosomes. That are the physical basis. The physical basis for the inheritance is chromosomes. So in examination also, what is the physical basis of the inheritance is that are the chromosomes, because genes the inheritance means the transmission of the character. So that transmission of the character is controlled by the genes. That genes are present on the chromosomes. Similarly, the Boveri also observed some of segregation, some of the separation of the chromosomes during the gamete formation in sea urchin. So these both are proposed this theory independently. So independently, and next the after the evaluation of this theory, these both scientists, we will consider that these two scientists propose this same theory. That's why that is the chromosomal theory of inheritance. I already told, according to this theory, the genes are present on the chromosome that are transmitted to the parents to progeny. So that what Mendel proposed. And here the chromosome number for grasshopper is 2n is equal 24 or 23 ma. Because in grasshopper, male grasshopper and female grasshopper is there. For male for female grasshopper, it's having the 24 chromosome. For male, it having the 23, because it having the system of XXX0 system. The sex determination of grasshopper is by the XXX0 system. And coming to the Barbara McClintock, so he was the first lady got the Nobel Prize in the field of the genetics. So in the field of the genetics, that means he got the Nobel Prize in the field of generally they were given under the medicine only. So Barbara McClintock, so he first identified the jumping genes. He first observed the jumping genes. This scientist will comes under the comes under the molecular genetics, because the contribution from this scientist Barbara McClintock used in the mostly significance in the molecular genetics. But this one is most important for the objective type, because generally they will ask about the jumping genes. What is the meaning by the jumping genes? So jumping genes means some genes, not all genes, in some genes in the DNA, in the chromosomes of the nucleus will move to one place to another place. That means one chromosome to another chromosome. It will be jump. These genes are jump one chromosome to another chromosome. That is called jumping genes, or transposable elements, or transposons. So jumping genes also called transposable elements or transposons. So keep remember this one. It was discovered by Barbara McClintock in the maize crop. In maize crop. So in maize crop, and the chromosome number for maize is, how many chromosomes are present in maize? 20. Okay, ma. I hope you understand this one, ma. Yeah, 10 linkage group. Okay, ma. Harsha Patel. Okay. So Harsha was raised one thing, that is the maize having the 20 linkage groups. Linkage groups. Generally, maize crop having the 20 chromosomes. That's why the linkage groups are 10, because the linkage groups are equal to the number of the haploids. Okay, ma. So tell me, how many groups are present in the humans, ma? How many linkage groups are present in the humans? Okay, ma. In case of the male, how many groups are present? In case of the female, how many groups are present? Ma Bhagya Lakshmi, ma in humans, there are the 46 chromosomes are present, ma. Okay, ma 23 and 24. In which
Yes,
case?
yes, sir. That only I was saying, but I wrote it. Means
Okay, okay, ma. I understand. So, ma Aarti, in which case 23? In which case for 24, ma? In male 24. Okay. In female 23, female 23, female 23. Why, ma? How? Okay, ma. Can I explain this one, ma? Yeah. Because generally in animals, so in generally in animals, there are the two types of sex you can see, ma. That is the one is the male and another one is the female. So that's why we have the two types of the chromosomes. One group is the autosomes, another one is allosomes. In humans, generally we have the 46 chromosomes. If you'll see in the male, there are the 44 chromosomes 44 chromosomes along with 1x 1x plus 1y. So generally, and if in female, there are the 44 chromosome along with 1x plus x. So here you can see the, so in heterogametic individual, you can see the number of the linkage groups equal to n plus 1. That's why the haploid chromosome number from autosomes is 22, and this having the one linkage groups and this chromosome having the one linkage group, because this are two different, that are the heterogametic type. That's why we will get the 24 chromosomes. So 24 chromosomes. But in case of the in case of the homogametic, because in some cases the male having the XX and female having the XY in some cases, ma. But here we will see anyway the chromosome number for the heterogametic, which one having the heterogametic is equal to n plus 1. That is a haploid chromosome number plus 1 for the heterogametic individual. For the homogametic individual, it is equal to the n only. I hope you understand. The linkage group for the heterogametic is the linkage group for the heterogametic is n plus 1. N means number of the haploid chromosome number, 1 is addition to that one, and the haploid the linkage group for the homogametic is equal to the number of the haploid chromosome number. And coming to the Benger-Seymour, so he's also done extensive work on the genes, the molecular level of the genes, and he has given the definition to the gene is entirely differ from the convention, because if you will see the evolution of the gene, if you will see the evolution of the Mendel has thought that the some factors, he don't know what is that factor, how that factor will made. And coming to the Morgan, Sutton and Boveri, Hugo de Vries, they don't know about the only they know this some gene, term is the gene. They don't know what how the gene will form, and how the gene will act, and how the gene will express. Only they were proposed these are responsible for character, these are transmitted like this one, and some of the exceptions also identified. So only this phenomena is there about the genes, and the concept of genes gradually changed, because earlier a character is controlled by the gene. According to the Mendel, a character is controlled by the gene that are controlling that gene having the two alleles. And later, by the Sutton Boveri, the genes are present. The two alleles of the genes are present in the two homologous chromosomes. They were proposed, and laterly, how the expression of gene by the Sutton and Boveri, they proposed that a character is expressed by the proteins that are formed in the form of the one gene is produced the one enzyme. According to Sutton Boveri, one gene is produced the one enzyme. That enzyme will controlling the character, and after that, one gene and one polypeptide by the Ingram that scientist also is there. So he has proposed the all the genes are not produced to the one, all individual genes are not produced to the one enzyme. So only some gene having the that type of the expression, but in most of the cases, one gene only express the one polypeptide in the enzyme. That means one polypeptide in the protein. So he proposed that, and he has given the explanation with the experiment also. That is the Ingram. So the concept of the gene gradually changed. So we came from the the changing of the concept of the gene is changed from the physical form to the molecular level. Now we are seeing in the gene, we are seeing in the nucleotide level. So that is a SNP, single nucleotide. So we are seeing at the level only. So similarly, the Benger also given the concept of the gene. That means earlier only one gene is controlling the one character. That means only gene is the made up of the nucleotides. That gene totally express the one character. But according to the Benger, one gene having this subdivision also. That is one is the cystone, another one is muton, another one is the recon. He proved like this. So he has worked on the T2 bacteriophase of the T4 bacteriophase in the R2 locus. This is because this is the genome of T4 bacteriophase. Bacteriophase means which are the viruses or live on the bacteria is called bacteriophage. So this T4 bacteria, this is the T4 bacteria, it is live on the E. coli. So this having because viruses having the only genome and protein code. So generally they have the only maximum 2 genes or 3 genes, because they don't need the all much genes, because this genome having the genome size will be comparatively high, but the active genes only 3 or 4 genes, because they don't need that much genes, because they don't need to take the food, they don't need to take the breath, that is the respiration. So only they have to attack the one individual, and they have to reproduce. That's why they hardly having the 2 or 3 genes, ma. So this is the active regions of the this T4 bacteriophage in that R2 locus. So he has observed the within the gene, within the gene also you can dividable. Earlier before the Benger, the gene is the individable, ma. So you cannot divide the gene. But after the Benger, gene also dividable. So each gene having this subunits, the cystone, muton, recon. The cystone will express the character. Muton will mainly involved in this region. This is the muton also the subdivision. This is the region of the gene mainly involved in the mutation, and the recon. It is the mainly involved in the recombination. So these are the 3 subunits, and we will discuss in detail about this one in the chapter of the concept of the gene. So you keep remember who was given the term, who was divide the gene into cystone, recon, muton is Benger. And this table, it is present in the Pundan Singh. This one is most important for the examination purpose. You should remember and you should learn about this scientist. Here I will go through briefly. First one is Morgan. He has proposed the gene theory and linkage theory. He has done he has done extensive work on the white eye color in Drosophila. So he proposed the linkage theory and gene theory. And Muller. So after the Hugo de Vries, the Muller and Standler, these both are induced these both scientists were induced the mutations in artificially. So in Standler, he was induced the mutation in barley, and Muller he was induced mutation in Drosophila. So you remember both Standler and Muller, this both scientists you get in the plant breeding also, and Beadle and Tatum this one is all of you know, and Watson Crick, this one also you know. Jacob and Monod proposed the Operana, and this scientist, you don't remember this scientist because these are not important for the examination. Only scientists, not years. So only some years are important for the examination. That is the when was the PCR introduced, and in which year PCR was introduced. So that one important. So only some years are important, and other than that important years, there is no need to buy out this years. But we have to remember all the scientists. And Nierenberg, Holy, and Corona, they were proposed the genetic code, and they are explained about the protein synthesis also. And this is the most important one, Temin and Baltimore. This one is important for the examination. Temin and Baltimore, who was given the process of the reverse transcription in tumor inducing viruses, who was given by the scientists of Temin, Baltimore. That's why this reverse transcription also called Teminism. The reverse transcription also called Teminism. So this process was observed and discovered by the Temin and Baltimore. What is mean by the reverse transcription means? Generally, the DNA will give the DNA will produce the RNA. That RNA will produce the protein. That protein mainly involved in the expression of character. Generally, DNA will give the mRNA. In some cases, in some viruses, this process will be reversed. That means that mRNA will convert it into the DNA. That is called reverse transcription. We will discuss in detail about that one in molecular genetics. But you should remember this scientist, Temin and Baltimore. They were discovered the reverse transcription. So that's why it is called Teminism. The reverse transcription also called Teminism. And the enzyme involved in reverse transcription is reverse transcriptase. And Barbara McIntosh, he discovered the jumping genes. And Phillips and Roberts. This one also important for the examination. Phillips and Roberts. These are discovered the split genes. So we will discuss about this one in coming classes. And Carey Mullis. Carey Mullis and Michael. These were discovered the PCR and directed mutagenesis. So these are the important. You should remember this one. And another scientist also there, Kornberg. He is also worked on the molecular basis of eukaryotic transcription. So you need to read this all the scientists and their contribution. So that is about today class, and you may have the quiz in next class about this class. If you have any doubts, you can ask. But you should remember, I only explain about the only superficial about the scientist and concepts, and I will give the much detail in the respective chapter. Any doubts, ma? No, sir. Okay, ma. I have one doubt, ma. That chapter is completed or not, ma? That one. I think Devdatta taken the class about the cell organelles, na? She has taken the class about cell organelles. How much she covered, ma? Plasma membrane. So, which portion she covered in cell organelles, ma? Plasma membrane means from the from the cell, we will start with the cell wall in plant cell wall. After that, plasma membrane. After that, we will go to nucleus, endoplasmic reticulum, Golgi complex, and mitochondria, chloroplast. So what she covered? Only plasma membrane she covered. Okay, ma. She covered nucleus. Okay. So you all people directly entered into the nucleus, without breaking up the plasma membrane and cell wall. You entered the nucleus. You have that much resistant, ma? Sir, ma, I'm just kidding. Okay, ma. So we will cover all the. So both are accepted, ma. The one gene, one enzyme is also accepted, and one gene, one polypeptide also accepted. Because if you see in the bacteria, one gene will give the one enzyme, one total enzyme it will give, ma. So yeah, but in case of the eukaryotic organism, the generally the enzyme will form by the proteins, and that proteins will form by the polypeptides. So in most of the eukaryotes, one gene will give only one polypeptide. But in case of the eukaryotic also, you can see the in some genes will act as the one gene will give entire protein. That is a monogenic character. That is it is example for the monogenic character. In case of the monogenic character, that means one character is controlled by one gene only. One character is controlled by one gene only. So that gene will give the entire, because the expression of character depends on the enzymes only, or some secondary metabolites. So if you want to express the any character, if you go to background of the character, first the enzyme will express, that enzyme will appear, that the expression of enzyme we are seeing the external appearance, the expression of enzyme only. We are seeing the physical appearance. So if you will take the any concept in the genetics, so that is not generalized. So the genotype of the each the gene, we are all having the same genes. We are all having the same genes, because maybe difference having the 0.0000000001%. But each genotype, each one is the differ from the another one, because of the so many factors are involved. Because someone will have the more resistant to something, and someone will have the less resistant, someone will have the medium resistant. Now we are seeing now resistant to the corona, because most of the people are got resistant, and some of the people are die, and some of the people are recovered without symptoms, with symptoms. Some of the people are they don't know they affected by the corona or not. They don't know affected by corona, but they got the resistant to corona. So each individual on the earth is the one genotype. Okay, next class we will start with the cell organelles. I will start from the starting. Cell wall, cell membrane or plasma membrane, and nucleus, and I will cover. I will come with the total cell organelles chapter, because we have to need to understand the genetics. So you have to understand the cell organelles, and mitosis, meiosis. These both are most important for the understanding of the genetics. One is the cell and cell organelle. Another one is the mitosis, meiosis. If you will understand these two concepts, you can easily understand the genetics, and you can easily understand the plant building also. You can easily understand the molecular genetics also. So mitosis and meiosis, and cell organelles, and DNA replication, and these three process: replication, transcription, and translation. You should need to know every step in this three process also, replication, transcription, and translation. So these concepts, if you will understand clearly, you can easily get all the concepts in the genetics. Okay, thank you for all, and have a nice day, Okay, Okay, I will take the next class on Wednesday, Okay,